Okay. Today we have with us Emmanuel Moebel. Emmanuel is PhD in image processing, and he works for the National Institute of Research in Computer Science and Control in INRIA, Renoir, Bretagne, Atlantic, France. He's going to present us DeepFinder. As DeepFinder is a novel neural network-based method to spot macromolecules in tomograms. And if in any moment you have questions or comments, you can write them in the chat. I will read them at the end of the talk, or alternatively, if you can wait until the end and use your microphone to ask directly, you can do it. So please, Emmanuel.
So, hello everybody. Do you see my slides?
Yes. Yes.
Okay, cool. So, welcome to my talk, and where I will present DeepFinder, a new particle picking algorithm for cryo-electron tomography, and during which I hope to convince you of the potential of this new tool. So, this work has been accomplished during my PhD under the supervision of Charles Quevran in the group Serpico at INRIA in France. INRIA is a computer science research lab, and the Serpico group focuses on computational bioimaging. So I will first shortly present you the context and the challenges of this work. I will then describe the method, and finally present some results. So my thesis has been developed in the frame of a joint collaboration between the Fourmentin-Gilbert Foundation, whose long-term objective is to create a 3D atlas of the cell, and the Max Planck Institute of Biochemistry, where I had the opportunity to work as a guest scientist in Professor Baumeister's group. So as you all know, cryo-electron tomography suffers from severe noise. The noise variance is 10 to 100 times larger than the signal. As a consequence, the cell structures and macromolecules are only light contrast changes. And as an illustration, here's a macromolecule classification task, and where it is very difficult to discern things with the naked eye. And therefore, we need robust computational methods to perform classification. Another problematic is that the cell is a crowded environment. So several macromolecule families are floating around and interacting with each other, which makes it a challenge to differentiate them. So to summarize, my challenge was to develop an algorithm for the recognition of similar objects within a crowded cell environment in very noisy 3D images, which may have reconstruction artifacts. The solution we proposed is called DeepFinder. It's a fast and robust approach for macromolecule identification based on supervised deep learning. It is an open source Python software. It has a graphical user interface, and it exists both as a standalone and as a Cyclone plugin. So DeepFinder is based on supervised deep learning. Therefore, it relies on data annotation. The method is based around image segmentation. So it can also be used to segment structures like cells and like cell membranes and microtubules. And it is by analyzing the segmentation map, which we do by applying a clustering algorithm, that we retrieve the coordinates of the target particles. So let's now talk about deep learning in general. So we chose to use deep learning for this task because it is clearly the state of the art in image classification, segmentation, denoising, and object recognition. And it has been applied to various fields, such as computer vision, language processing, and bioinformatics. And the increasing success of deep learning can also be observed in the cryo-electron microscopy field, where a growing number of methods are published since 2016. Deep learning has been investigated for particle picking in single particle cryo-EM, also for supervised and unsupervised segmentation, and for supervised subtomogram classification. Now, what sets us apart compared to these methods is that we provide a 3D multi-class deep learning framework, meaning that we can detect several particle classes at once. Also, the method is robust enough to perform successful detection in a cellular environment, which, as you know, is more complicated than in the single particle framework. In addition, we compare our performance to template matching and competing deep learning-based methods, thanks to the Shrek challenge, which I will present later. Finally, we evaluate DeepFinder on experimental data for several macromolecule classes with molecular masses as low as 550 kilodalton. So before describing DeepFinder in detail, I would like to make a short introduction to deep learning, as some of you might not be familiar with it. So first, let us define what an image filter is. A filter is defined by its kernel, which is an array containing values. So this kernel is combined with an input image with an operation called convolution. The output is a filtered image called feature map in the context of deep learning. Depending on the values in the kernel, different filterings can be obtained, as can be observed here. For example, on top, we can obtain blurring or enhance features like contours. Now that we have defined what an image filter is, let's define what a convolutional neural network is. So it's a neural network adapted to image data. A CNN is composed of several so-called convolutional layers. These layers can be seen as filter banks, where each bank can contain several hundreds of filters. By applying these layers sequentially, more and more complex features are extracted from the input image until a desired output is achieved. As an example, here at the bottom is an illustration of features encoded in a face recognition task. The first layer encodes basic features such as contours, and progressively, layer by layer, the CNN encodes eyes, noses, and finally the desired feature, the face. And in the end, a CNN enables a hierarchical representation of the data. So now comes a natural question. Which filters should I use for my application? And the good news is that you don't need to, because they are directly learned from the data thanks to our training procedure. So the training procedure has two inputs. I think there is a...
Sorry, Jose Maria. Jose Maria, can you mute? Yeah, thank you.
Okay. So the training procedure has two inputs. On the one hand, you have the training data, and on the other hand, you have the annotations, which are also called training targets. And these targets represent the output you would like to observe. First, at the start of the procedure, the filter weights, so the values in the kernels, are initialized in a complete random manner. Then the data is processed with this random neural network. And at this point, the output of the CNN, which is called the prediction, will essentially be garbage. Second, the prediction will be compared to the target with a loss function. This loss function measures how far apart the prediction is from the target. And then the computed loss is passed onto our optimization algorithm called the stochastic gradient descent. The role of this optimization algorithm is to update the filter weights in such a way that the loss score decreases. And then, by applying this procedure iteratively, the loss should decrease and reach a minimum. At this point, if the training is successful, the output should be identical to the targets and the training is accomplished. So let's now pass to the description of the DeepFinder algorithm. So the neural network we are using here is based on the UNet. The UNet is a network that has been designed for image segmentation. We achieve optimization using the Adam algorithm, which has been chosen for its good convergence properties, and the dice loss, which has been shown to behave good in segmentation tasks. We also had to adapt the training procedure to be able to run on large 3D volumes. Due to memory limitations, it is not always feasible to load the whole tomogram set. And therefore, we sample smaller 3D patches. Moreover, in cellular tomograms, there is a severe class imbalance. Only 0.5% of voxels are occupied by macromolecules, meaning that the negative class, the background, is overrepresented. This perturbs the training. The CNN only learns the overrepresented class and ignores the underrepresented ones. So to address this problem, we chose to sample patches only at annotated positions. In other words, we make sure that each patch contains at least one macromolecule. So let's now describe the workflow of DeepFinder. So we have two stages, the training stage and the analysis stage. And in each one of the main steps of the workflow, represented here by the blue boxes, the user is assisted by a graphical user interface. So first, in the training stage, the user has to annotate the positions of the macromolecules. 2, these coordinates are converted into segmentation targets for the training. And 3, the tomograms and segmentation targets are fed to the training procedure, and the training is launched. Once the network has been trained, it is ready to be deployed to analyze new tomographic data. So, as mentioned in the earlier, the user is assisted by a graphical interface. So here, the user can explore the tomogram with an auto-slice view. He can also denoise the slices for better visualization. And by clicking on the slices, he can annotate the positions of the macromolecules of interest. So once the coordinate list has been obtained, we have to convert these position-wise annotations into voxel-wise annotations, meaning the segmentation targets. And this is not a trivial task, which is why we propose 2 different strategies of doing so. So the first strategy is based on subtomogram averaging. So we use the annotated positions to extract 3D patches, which are given as an input for the subtomogram averaging procedure. This procedure gives us 2 outputs, the subtomogram average, which we can use to generate a binary mask, which encodes the 3D shape of the particle. And the second output is the particle orientation. Now we can copy-paste the binary mask into an empty volume at given particle positions and corresponding orientations. The result is a fairly accurate segmentation target. So here, the subtomogram averaging is applied on binned data. So the computing time is not as high as usually. Nevertheless, it can still be 30 minutes to 1 hour, which can be a bit long. Therefore, we propose a second strategy, the sphere-based targets. So here, instead of using the exact shape of the particle, we simply use a sphere mask. Therefore, the orientation is not needed, and subtomogram averaging does not need to be applied. And therefore, the computing is much faster, only a few seconds. The resulting segmentation target is an approximation and not as accurate as before. Nevertheless, we have shown that in our paper that in practice, we obtain a very similar performance compared to shape-based targets. So, once the segmentation targets have been generated, the training can be launched. The graphical user interface displays in real time the evaluation metrics, which allows to monitor the progress of the procedure. Once the score curves reach a plateau, the training is done and we are ready to analyze new data. So now that I have described DeepFinder, let's present the different data sets we have evaluated DeepFinder on. So the first data set is a synthetic data set, and the 3 others are real data sets with, for each one, a focus on a different macromolecule. For evaluation, we will use the F1 score, which is pretty common in detection problems. And it can be interpreted as a harmonic average between the recall and the precision. So the first data set is actually a benchmark that has been created for a challenge organized by the cryo-electron microscopy group of the Utrecht University. It contains 12 classes of macromolecules with varying sizes. The dataset has been designed to show the breakpoints of particle picking algorithms, which is why some of the particles are very small. The training set is composed of 9 tomograms and the test set of 1 tomogram. So in 2019, a total of 8 research groups have participated. One of them used template matching, and all the others used deep learning-based methods. And I was very happy to discover that DeepFinder was the clear winner of the 2019 edition. Let's now have a look at the scores per class. So here in blue, we have the scores for DeepFinder. In red, we have the scores of the closest competitor. And in green, as a reference, the scores for template matching. And as you can see, for all methods, the scores are correlated with the particle size. The smaller the particle, the harder it is to detect. Template matching can achieve competitive scores for larger particles. However, it is particularly bad at detecting smaller ones. On the other hand, DeepFinder is doing quite well. While the scores are worse for smaller particles, it is still better than competing methods. So in this slide, I would like to show the influence of the training set size on the detection performance. On the y-axis, we have the F1 score. On the x-axis, the number of training tomograms, knowing that in each tomogram, there are approximately 200 particles per class. So what we can see here is that for small particles, the scores drop pretty fast. But for larger ones, the score is remarkably stable, even for training sets composed of only one tomogram. So clearly, the required training set size depends on the particle size. And actually, as is common for segmentation with deep learning, the determining factor is the number of voxels occupied by the particles. Therefore, in order to detect small particles, we need more annotations than for larger ones. Let us now move to real datasets. So in dataset 2, the focus is to detect ribosomes. Ribosomes are large compared to other macromolecules, and therefore it is considered a bit as a textbook case. So when you develop a particle picking algorithm, it is generally the first challenge that must be met. So here, we train DeepFinder to detect two binding states of the ribosome. Membrane-bound, in blue, and cytoplasmic in yellow. We also added the membrane class to illustrate that DeepFinder can also be used for classic segmentation of cell structures. So this dataset is quite large and ideal. We have 48 training tomograms, and experts annotated around 6,000 ribosomes per binding state. First, let us take a look at the score maps. So on the right, we have the score map produced by DeepFinder. And in the middle, a score map produced by template matching, which is a common choice for particle picking. And as you can see, the template matching score map is very noisy. We have high score values at particle positions, but also at everything that has a similar contrast to the ribosome, like, for example, the membrane. Therefore, it produces a lot of false positives, and post-classification techniques have to be applied in order to decrease the false positive rate. On the other hand, the DeepFinder score map looks much cleaner. High values of scores are organized in well-shaped blobs, and therefore the scores produced by DeepFinder are more discriminative than the ones used by template matching, which is the constrained cross-correlation. So in the end, DeepFinder does not need post-classification like template matching does. And finally, this is the kind of segmentations that we are able to achieve. Visually, the segmentation makes sense. The detected membrane-bound ribosomes are in blue and are always located against a membrane, which, by the way, was also segmented by DeepFinder. And in yellow, there are the cytoplasmic ribosomes. So here, we quantify the detection performance by computing the F1 score with respect to the annotations. So in blue, we have the score for DeepFinder, and in yellow, the score for template matching. DeepFinder achieved a score of 0.86, which is pretty good, while template matching only achieved 0.5. Actually, template matching is usually pretty efficient for detecting ribosomes, but here, the task was to differentiate between the binding states, and therefore, template matching was confused between the two classes, and hence the lower score. We also evaluated the obtained subtomogram averages by estimating the resolution. We computed one subtomogram average from the expert annotations and another from the DeepFinder detections. And as can be seen on the Fourier shell correlation curves, both reached a similar resolution. However, the DeepFinder average resolution is a bit lower. Possible reasons could be that DeepFinder has detected false positives, but it could also be that the detected ribosomes are more noisy or have more structural variability, which would blur the average. So to investigate this question, we have studied the complementarity of the expert set, here in blue, and the DeepFinder set, here in yellow. Both sets have a large overlap, but Also, they have complementary parts, which are unique to each set. So the question here is, what is the nature of the extra detections that DeepFinder made that are not part of the annotations? So to answer this question, we decided to compute the subtomogram average from these extra detections. If the resulting average contains only noise or some kind of strange blob, then we can conclude that these extra detections are false positives. However, this is what we obtain. So we can clearly observe a ribosome density. And therefore, we can conclude that a majority of the extra detections are true positives. And as a consequence, thanks to DeepFinder, we could increase the membrane-bound ribosome set by 20%. So this experience also highlights the value of using several particle picking techniques. By combining their detections, a more complete particle set can be obtained. Let us now move to the next datasets. Starting from here, the results I present have been obtained very recently. In dataset 3, the focus is to detect Rubisco holoenzymes. This task is more challenging than before because a Rubisco particle is much smaller than a ribosome. Here, the dataset is composed of only 4 tomograms. However, the number of annotations is huge, almost 130,000 particles. These tomograms are basically oceans of Rubisco complexes. So when comparing to expert annotations, we obtain a F1 score of 0.83, which is similar to a similar value to what we obtain with the ribosomes, even though the Rubisco complex is much smaller. We also compared the obtained subtomogram averages. So in blue is the one obtained by the experts who use template matching and CPCA classification. And in red, the one obtained with DeepFinder. And as can be seen on the Fourier shell correlation curves, the achieved resolution is quasi identical. Let us now move to the last datasets, where the focus is to detect photosystem 2 complexes. This dataset is the most challenging because PS2 is small, and also because it is a membrane protein, which is more difficult to detect. And an additional difficulty is that here the training set is very limited. It is composed of only 3 tomograms and only 298 particles. Here, the experts had to annotate the data per hand. The reason is that template matching was very inefficient for this dataset, as can be seen on the template matching score maps here and here. And in contrast, the DeepFinder score maps are more precise. So next, we have computed the F1 scores obtained for different membranes of the test set. In blue, we have the scores for DeepFinder, and in red for template matching. And as you can see, the scores for template matching are limited, and globally, they are 2 times lower than DeepFinder. The DeepFinder scores vary a lot with respect to the membrane, from 0.18 to 0.74. And we observe that the low scores correspond to poorly resolved membranes. Nevertheless, we believe that this result is promising because the data set is limited. We are convinced that adding more annotations to the training set will improve the DeepFinder performance for PS2 complexes. And also, this result demonstrates the potential of DeepFinder to detect membrane proteins. Now that I finished presenting the DeepFinder performances on our 4 datasets, I would like to speak about generalization potential of DeepFinder and of deep learning-based techniques in general. So here, in this image, the training sets and the test sets do not originate from the same dataset. DeepFinder has been trained on tomograms of chlamydomonas cells and applied on tomograms of P19 cells. Nevertheless, DeepFinder was able to detect membrane-bound ribosomes in the P19 cells, even though these cells do not originate from the same species. In one case, we observe alga, and in the other, we observe a mouse. So this figure represents a preliminary result, which can help us imagine the future of cryo-ET analysis. We can imagine that in the future, the biologists could have access to a bank of pre-trained general-purpose neural networks ready for analyzing new data. Here, the biologists would not need to execute the annotation and training themselves. It would have been done beforehand by other research teams. So transferability of neural networks in cryo-ET is still an open question. How similar should the trained sets and the test set be? Should they have the same voxel size, a similar signal-to-noise ratio? Should they have a similar tilt range? Well, we are not sure now, and future research will allow us to unlock these questions. I also wanted to shortly mention the computing times of DeepFinder. So while it is true that the training can be long, a few hours to 15 hours, once trained, DeepFinder only needs 25 minutes to analyze a tomogram, which means that dozens of tomograms can be processed in a single day. And to finish, here's a screenshot of the new DeepFinder plugin, which runs in the Scipion ecosystem. It has been developed in collaboration with the Scipion team and should be made available shortly. Also, my colleague from the Scipion team, George Jimenez, made a series of video tutorials, which you should have soon access to. So, thank you for your attention, and I hope I have attracted your curiosity and that I could convince you of the potential of DeepFinder.
Thank you, Emmanuel. It was a really nice talk of a really nice algorithm, and I hope it will be published soon. There one question in the chat. You can read it if you want. I will read it anyway for all people. So Philippe Schoenbeck asked, you said you only use patches for training which contain macromolecule labels. Does the network have problems later after the training when confronted with empty patches? And if so, did you try to also add some empty patches for training?
So it is true that when you train a neural network, negative examples are as important as positive examples. And also because the negative class can have much more variability. But in this case, the patches are large enough to include enough negative examples around the macromolecules. So it was not necessary in my datasets to sample patches with only negative class. I hope that answers the question.
Okay. Thank you, Manuel. I see people is typing. Yeah, please go ahead.
Meanwhile, I got a question. Emmanuel, is that challenge data set is publicly available? I think it's a nice, it seems like to be a nice data set, the one you use for the challenge.
Yes, it's publicly available. There has been a 2020 edition and lately a 2021 edition. So each time they update the data sets to make it more realistic. So if you develop particle picking methods, I definitely advise to use this dataset.
Okay, I didn't know about it. Thank you.
Okay, I have one question for you, Emmanuel. Have you thought about the possibility of, because if I understood well, you say that the DeepFinder is template free. So have you thought the possibility of combining a kind of template free with a template? I mean, because perhaps if you provide a template, you train the net with a template, it helps a little bit, or something. It's a black asking, but
well, DeepFinder for training needs, on the one hand, the tomogram, and on the other hand, a segmentation target. So we have to. So when you speak about including a template for training, the only way I see where we can include the template is to annotate. I presented a shape-based segmentation target to annotate the macromolecules with binary masks of their shape. Okay. And we could use templates from the PDB databank to annotate our segmentation maps.
Okay. No, I was thinking if a kind of combination of template matching and deep learning, it can enhance the picking. But
another way of combining the techniques, which has been done, is you can generate annotations, the initial coordinates for the training sets, using template matching.
Okay.
This is actually how the annotations for the ribosomes have been obtained.
Okay, thank you. And Mohamed Rastani asked if starting from scratch, how many particles should be annotated on average before the network can start generalizing? Or what is the percentage of particles that should be found manually before the network can find others?
So as I showed in a slide, this depends on the size of the macromolecule. But to have a really good performance, I noticed you need around at least 1,000 particles in your training set. At which point the DeepFinder is able to find new ones. And then what you can do is check the new ones, because there will probably be false positives, because the initial training set is small. And then you can validate them and add them to a new training set. So you can make a kind of iterative curation, where DeepFinder proposes new detections, and you validate them or not, and retrain DeepFinder and make it iteratively. And I have done this with a team from Strasbourg, and we started with an annotation set of 400 particles. And quickly we were able to reach 1,000.
Okay.
So I hope this answers your question.
Yeah, I think so. Lin Huatai asked, thanks for your excellent talk. I have one question. Do you think it is possible for DeepFinder to assign initial orientation to pick it out particles while picking?
You mean that DeepFinder generates the orientation angles from the particles, like template matching does?
Perhaps, yeah.
Okay. Well, it was not the objective of DeepFinder to produce orientations. The objective was to produce coordinates, central point of each particle. But I imagine that yeah, maybe with a correct neural network architecture, it could be done, but this is a subject for new research.
Mm-hmm.
It's not in the frame of DeepFinder.
Thank you, Manuel. Philippe Schoenbeck asked if do you adjust voxel pixel size of the testing tomograms if it differs from the training dataset, or are you still testing this?
For now, I had always the same voxel size between training set and test set. I have not tried varying this parameter. But I have observed that DeepFinder is able to segment things when the voxel size varies a little bit. But I was not able to analyze this influence clearly for now.
Okay, thank you, Manuel. Koray asked the idea of use a spherical mask as ground truth for training in the segmentation problem is interesting. Have you thought about training a model to estimate the sphere center and radius from a patch? For instance, a regression-based model?
No, I don't. I didn't do that because in my case, well, the sphere center is what is produced by the person who annotates, and also the radius of the sphere is most of the time it is known because the biologist has an idea of how large the target's macromolecule is. And the sphere radius corresponds approximately to the macromolecule radius.
Thank you, Emmanuel. More questions. Mohamed Alastani asked, have you tried DeepFinder on data where only one particle exists from an in vitro or in simulated tomogram?
So you mean just one particle class, like in single particle, I imagine. Did I actually? No, not. I have not tried that. Although the Rubisco dataset is pretty similar to this scenario, because it's basically full of Rubisco particles, and in this scenario, as it is, the scenario is more simple than in a cellular environment. So I believe DeepFinder can be successful.
Okay, thank you, Manuel. I think there are no more questions. So
yes, I have one. I was thinking about the question José Luis Vilas has made before, and continuing with this specific question, is it possible to use some kind of particle which has been reconstructed after a subtomogram aberration workflow to then feed again a DeepFinder just to make it look only for one kind of particle, only this specific shape, like a template matching, but more specific to the project you are working with?
Sorry, can you reformulate your question?
Yeah. Talking about the combination of template matching, I was thinking if how complex can be that kind of template. For example, if I'm getting an initial model from a subtomogram reconstruction, can I use this information to feed again DeepFinder and get a refined kind of picking some way?
Well, I think yes, you can use a new, more refined model average to generate more precise segmentation targets.